Hello everyone, my name is Anwar, glad to see you on my Bioculture channel and welcome to my SAT biology course. If you haven't subscribed yet, then be sure to be subscribed today, press like and share this channel with your friends and colleagues. Today we'll consider the second type of macromolecule, lipids. So before we begin, let's dwell on what lipids are. Lipids are one class of large biological molecules that do not include real polymers, and they are usually not large enough to be considered macromolecules. However, they are grouped with each other because they have one important feature. They mix poorly, if at all, with water. Here we will focus on three main types, namely fats, phospholipids and steroids. So, fats. They consist of two main structures in a ratio of 1 to 3. The first structure is glycerol. It is worth noting that the ending OL means alcohol. The second structure is fatty acids. They have a long carbon skeleton, usually 16 or 18 carbon atoms in length. In them we distinguish two regions. This is the carboxyl group, CO, and the hydrocarbon chain. You might think, how do glycerol and fatty acids bind together? As you can see, glycerol has an OH group on each carbon. Also there is OH in the carboxyl group of fatty acids. It turns out that everything happens like this. Hydrogen is taken from glycerol and the OH group is from fatty acid, so that a condensation reaction occurs with the release of water. I think everything is clear here. Let's move on. You should also consider that the fatty acids themselves are divided into two subgroups, saturated and unsaturated fats. Here I will tell you only about the things that you should know. In general, unsaturated fats have a double bond, a kink in the structure, and cause the fats to be liquid at room temperature. For example, sunflower oil. And as for saturated fats, they have the opposite. In other words, they contain only single bonds, straight, and lead to the fact that fats are solid at room temperature. For example, butter. The next class is the phospholipids. Here only a couple of things you should know. The first is that they are composed of glycerol, two fatty acids, and a phosphate group. There is also an additional charged or polar group that is attached to the phosphate group. For example, in this illustration it is choline. The second is that we distinguish two regions of phospholipids. Namely the hydrophilic head and hydrophobic tails. In other words, one of the parts of the phospholipid either expresses an affinity for water or conversely avoids water respectively. And third, you should know that phospholipids are the main components of the membrane. So let's move on to our last aspect of today's video, namely steroids. Steroids are lipids that are characterized by a carbon skeleton consisting of 4 fused rings. Here you should know examples of steroids. For example, one of the common ones is cholesterol. A couple of facts about this compound. It is a common component of animal cell membranes as well as a precursor to other steroids. In vertebrates, cholesterol is synthesized in the liver and also obtained from the diet. Also, it is very important to know why there is a variety of steroids. The answer is simple. This is all due to the variation of chemical groups that may be part of the molecules. Okay, on this our fourth lesson ended. As usual, I will leave a link at the description to download today's slide. I hope it was the most useful for you. And if you haven't watched my previous videos, be sure to check them out. I will be waiting for you in the next lesson. Всем большой рахмет.